I would like to start by thanking my nominators, Professor Wendy Nelson and Professor Marty Anderson, for nominating me for this award. And I also would like to point out to the Royal Society that many people, including myself, would not have been nominated for a fellowship without being really pushed to do so by my two mentors, and that I am very grateful for them and having the confidence to elect me as a fellow, or you to nominate me as a fellow. So I have been elected as being at the forefront of research on impacts of anthropogenic change on life in the ocean, and mainly I work on embryos and larvae. And we're all familiar with things like this. Things are pretty terrible in the earth at the moment. We've got increased CO2 concentrations in the atmosphere. The oceans are a big sink for all of that CO2, and it has impacts on lots of things, including ocean acidification, lowering the pH, changes to salinity with melting of the ice, and increased sea temperature because of increased air temperature. And myself and my laboratory have been working on this for a long time, some of it funded by the Marsden Fund. And basically, the take-home message is it's sometimes neutral, mainly bad, sometimes very bad. But today is about celebrating life and the success of all of our fellows. So instead of telling you about the impacts of anthropogenic climate change on the oceans, I'm going to talk to you about larvae, which are much more interesting. So what is a larvae? Well, a larvae is the stage in the life cycle. So most marine invertebrates have a larval stage. So after we go through the early part of development at fertilization, we become a zygote, and then we go through our embryo stages. How do I get that little thing? Oh, here. So we go out through our cleavage stages, 2, 4, 8 cell stage. All animals go through a blastular and a gastrular stage, and then we develop a larval form. Now in this cushion star here, Pteriala regularis, which if you've been to the intertidal in New Zealand, it's found pretty much everywhere. You've probably seen that. Its larval form is a biponaria shown here, which gets to after a couple of days, and then after it stays in that stage feeding on the plankton for 4 to 6 weeks, and at the very end of its larval life it comes down to the bottom with the small rudiment of a starfish that develops on the left hand side, and then that, like a lunar landing module, comes down to the bottom and becomes the little starfish. So every marine invertebrate has this part of this life cycle. It's not just too dissimilar from what happens in, say, your caterpillar developing and going through a process of metamorphosis to become a butterfly. Now, most of the marine invertebrates that have larvae, the larvae are found in particular class or phyla, but they don't look anything like the adult. And so you can see here that the adult starfish, as shown here, it only has a star form when it gets to this very advanced stage. So there's quite a lot of knowledge about what the larvae of different things look like, so the morphology of that. So early on in my career as a postdoc in Craig Young's lab, who was at Harbour Branch in Florida at the time, we put together a book called the Atlas of Marine Invertebrate Larvae that was compiled photographs and line drawings of all of the larval forms in the ocean. This was partly done to celebrate the retirement of our joint PhD advisor, though we were different generations. He was early generation, I was later generation. And this book was published in 2002 and is out of print and highly valued. Second-hand copies on Amazon go for over $500 US, so that's kind of good. I've got quite a few on my bookshelf actually, so at the university. Yeah, easy. And we're just doing now the second edition, which hopefully will be available this year. One of the other parts of my research program is to identify to get them to species level, we take advantage of DNA barcoding, because of course it's part of the life cycle. So a larvae grows into a juvenile, which goes into adult, so it has the same DNA sequence for any particular gene as it does. So you can match those adults and their larval forms. So this is some work that we did in Antarctica. And in the middle, we've got the larval forms, and on the side, we have the adult forms. The ones with the pink boxes, we've been able to identify to the species level. Those on the left-hand side, we have not. We know it's a starfish, and we know it's a Nemertean Palidia, but we don't know which species it belongs to. And part of that is an issue with the databases. When we were doing this research on Antarctica, we discovered that many of the larval forms were in the plankton for over 6 to 8 months, and we wondered how they were managing to do that. So that took us all back to looking at some of the energetics of this, and we went into the biochemistry of looking at lipids and proteins. Going back to Rod's talk about triglycerides, one of the things that animals use for energy is your triglycerides. And so we used this instrument here, which is called an H-scan, which basically allows you to differentiate between the different lipid classes. And here the triglyceride, that's one of the bad lipids in your bloodstream, was really important for development. And we talk a lot in science about impactful research, and this is probably my sort of cringe and embarrassment, but I don't really, because this is the piece of research that is my most cited paper. Was on a paper called Contributing to Marine Pollution by Washing Your Face. And instead of being a long reflection or use of imagination, it was a small summer project that I did with a student, where I realized that all of these facial cleansing products, scrubs targeted at teenagers for removing acne and blackheads, were containing pieces of plastic which were about the same size, we discovered, as the food that the plankton were feeding on. And instead of these products were called microbeads, but in fact, if you look at the lower picture here, you can see that they're actually kind of little bits of fragments, and some of the other products, they kind of look like things you would clean off the floor. They weren't round and beady-like at all. So this paper was published in Marine Pollution Bulletin. It got a lot of media attention for a while, and it was picked up by NGOs, including Greenpeace and Ban the Bead, etc. And this actually led to the Waste Minimisation Microbeads Regulations of 2017, which actually banned the sale and manufacture of these products in New Zealand, and probably saved the ocean from quite a bit of plastic going in there. So here's a little bit of research that turned into a change in the law, so something to be a little bit proud of, I think. Here is one of the last stories I tell you, the mystery of the hyaline spheres. These are these strange structures that appear, the HS on these, in the arms of a sea cucumber larvae. And with a particularly talented PhD student, we were able to identify that the phytoplankton food being taken up by the stomach, this is actually a very big circle, went into these lipid transfer cells and then to the hyaline spheres. So these actually were lipid stores that were being taken through development. And in that graphic below, you can see that they go through from the auricularia stage, they go into the second larval stage, which is called the doliolaria, and they still are retained in the juvenile. And this was a mystery, and that had been first discovered in the 1880s, and it took 150 odd years for us to find out actually what they do. So in my talk today, although I have been nominated for my climate change research, I'd rather be remembered for the work that I do on larvae. And I've got two take-home messages from today: that larvae are important in the life cycle of most marine invertebrates. They are ecologically important because they link the animals that live on the benthos and they provide dispersal in the water column. They're very, very vulnerable to climate change, and they're very beautiful too.
Well, thank you very much for that talk. I'm sure there are some questions, so fire away. Katie's making up for the fact that she couldn't.
Yeah, I know, she is.
How big are they? What's the size distribution?
This one is a mega larvae, which is actually about 2.5 millimeters in size. This one is about 300 microns, so 0.3 millimeters. So mainly for young people, they're very visible to the naked eye. For old people like us, not so visible. But there are some larvae. This one here is 10 millimeters. So this is one of the super unknown larvae. A larvae we don't even know what it is of. We know it's a sea cucumber, but that's it.
And do things eat them? Are they a food source for somebody?
Yes, they are eaten by other larvae as well. Yeah.
We tend not to, but you could.
Yeah. Yeah.
Mary, can I ask you a deeply unscientific
Yes,
question?
of course. I'll get my imagination ready.
So, as you said, when the slide appeared, I thought, what absolutely exquisitely beautiful things. But can you just give us a little bit, I think you've hinted a little bit more of a sense of how you relate to larvae. Like, are they living, like, do you talk to them? Are they, I mean, you spend a lot of time with them. So
Yes, I do.
So
and
what's the relationship of a marine biologist to the species that they really love?
well, when you're actually culturing them and studying them in the laboratory, there's kind of like people who are known for having a green thumb and people who kill all of their cultures. So there are a number of people who sing to them. When you're trying to get the eggs and sperm to make your larvae, various people believe Barry White is quite useful in getting your animals to spawn. Other people go for the more classical route, but yes, it is that they are beautiful organisms, and I always feel a bit guilty when I have to kill them to do biochemical assays. But yeah, they inspire a lot of art too as well.
One more question.
And many people who study them have them as tattoos, not me, but other people do.
Thank you for a very interesting talk. There can be very many larval stages in the life cycle of one organism.
Yep.
And often the larval stages look completely different from each other. So how do you actually find out what belongs to what? You know, what are the actual larval stages of, a particular...
Well, as I said,
that?
you can use the DNA barcoding approach now, but otherwise what you have to do is you have to get eggs and sperm together and take photographs of what they all look like. But, as pointed, nobody knows what this is. We know it's a sea cucumber. We know these little white dots on here mean that it's from one of two families, but that's a long, long way. We know that it must be a widely distributed species because they're found all over in the Atlantic and the Pacific Ocean. But these are the mysteries, just like with the Highland spheres. They grab your imagination and you have to just keep, how can I answer this question? And so what I've done here with this one is there is a group on Facebook called the Blackwater photos. They take photos. So these are like semi-crazy people who go out in the middle of the night into the ocean, like the deep ocean, like in the 700 meters deep, and they float around in the ocean with their cameras taking photographs of larvae like this. And so I have put out on social media, anyone who sees this, please send me information on where you find them, when you saw it, how big it was, and things like that, because there's a lot of meeting of the minds in terms of citizen science helping us answer those questions.